Hello, everyone. Welcome to the HUPO ETC webinar series number 3. So this webinar series is organized by the Education and Training Committee under HUPO, and it's focused on clinical proteomics. So my name is Giuseppe Palmisano. Today, together with Shisha Wang, I will chair this webinar. So we have the pleasure to have with us Dr. Adriana Paes. So I will just give a very brief introduction because Adriana, she has prepared actually a slide for that. But Adriana, she's a group leader and the head of proteomics at the Brazilian Bioscience National Laboratory, LN Bio, in Campinas, in Brazil. Adriana, she did a graduation, master, and also PhD at the Unicamp, University of Campinas. And then she did her postdoc at Butantan Institute and the University of Virginia. So Adriana has a very bright career. She has been developing several methods for clinical proteomics using mass spectrometry, and especially studying head and neck cancer. So beside that, Adriana has a very big involvement in education. So she organized a proteomics workshop here in Brazil that is very successful. A lot of edition already happened. And also Adriana is a member of the Council of the Brazilian Proteomics Society. Before starting, of course, I would like to thank also HUPO for giving us the opportunity to organize and share these webinars, and especially the HUPO office that has provided us all the infrastructure for that. And before Adriana starts her talk, I would like to remember that we have HUPO 2025 this year in Canada. So please register. It's going to be an exciting event. So Adriana, I would like to thank you again for accepting our invitation. So the floor is yours.
Are you hearing me well, Giuseppe?
Yes, I can hear you.
Okay. Mm-hmm. So first of all, I'd like to thank you very much for the invitation. It's a great pleasure to be here and to present some results that we have conducted in the lab. And thank you very much to be part of this education and training webinars. So the title of this presentation today is Exploring Head and Neck Cancer from Initiation to Progression using Spatial and Single-Cell Proteomics. So before starting, as Joseph commented, I prepared this slide to show a little bit about my scientific journey. So I obtained my bachelor in dentistry, and since then I have been working with the features of saliva. I started mass spectrometry-based proteomics in my master's science and PhD, where I studied the proteins in the biofilms. And during my postdoc at Butantan Institute and at University of Virginia, I studied mainly the protease in the snake venom. And in 2009, I applied to a position at the Paes. And then I became a research and a mass spectrum leader in the facility. And I study mainly head and neck cancer, then oral cancer, focus on strategies and techniques based using mass spectrum-based proteomics. So the outline of my presentation, I will present a little bit about Paes LN Bio. I will talk a little bit how the spatial proteomics has been revealing diagnostic and prognostic signature markers for head and neck cancer, some initiatives for clinical translation from spatial proteomics, and then why we decide to move from bulk to single cell proteomics applied to biofilms. And then I will draw some perspectives. So CNPEM is a private nonprofit organization overseeing the Brazilian Science by Ministry of Science and Technology. CNPEM is responsible for managing the 4 national laboratories, and my national lab is here in the Brazilian Bioscience National Laboratory. CNPEM is responsible for connect these 4 national laboratories in these activities, open facilities, research and development, support for innovation, education, training, and outreach. Leme Bio is organized in the 4 divisions, and I am responsible for the advanced cluster in health technologies. The users, internal and external users, can apply their projects to the open facilities, and the users are not charged. It's open for both, are not charged to use the facilities. This is for both Brazilian and international academic users. The mass spectrometry lab is organized in 4 labs. We have the instrumentation lab, the sample preparation lab, the instrumentation lab, and also the data analysis lab. They're all open for the users to prepare their samples and learn mass spectrometry-based proteomics. We have 4 instruments dedicated for the internal and external users. We have instruments for discovered proteomics and also target proteomics. If you are interested in apply your project, you can access this website and submit your project. Specifically for mass spectrum facility, we are open for submission of projects in this month, and the proposal will be executed the following year after evaluation by external scientific committee. If you want to know more about Leme Bio, please, you can visit the Leme Bio website. So now we start talking about the spatial proteomics, how it can reveal diagnostic and prognostic signatures markers for head and neck cancer. But before starting that, I'd like to acknowledge the proteomics team. I am the voice here. They have been working hard over the years to review very interesting data that can progress in the studies of oral cancer and head and neck cancer, and also to develop new technologies based on mass spectrometry approach. And also thank you very much, the valuable collaborators. So it's important to mention first, why is it important to study oral cancer? The statistics shows that oral cancer is among the most prevalent cancer worldwide. And you can see here in this map, the high incidence of oral cancer worldwide. The risk factors associated with this disease are high consumption of smoking, alcohol, and the infection by HPV. Unfortunately, these patients have high rate of lymph node metastasis. I will show you several results that we using these clinical characteristics to study how can we anticipate the metastasis of the patients, because the lymph node metastasis associated with the main prognostic factors. These patients also have high rate of relapses, and the 5-year survival rate is less than 50% in advanced cases. Unfortunately, until nowadays, there are no robust diagnostic and prognostic markers available in the clinical practice, and the overall response rate of the available therapies remains low. So over the years, we have is exploring head and neck cancer and oral cancer to microenvironment through spatial proteomics, because different regions of the tumor can reveal molecular and histological characteristics associated to the clinical outcome. And therefore, we select the histology-guided proteomics mainly based on laser microdissection to map the tissue microenvironment and to review diagnose, prognose markers and therapeutic targets. So I am presenting here today many strategies that we have been using to discover and verify and validate the potential candidates. Before starting that, I would like to show a little bit our journey through bulk spatial proteomics. So in 2013, we evaluate the whole tumor in this study where we evaluate this in an orthotopic mouse model, the progression of oral cancer. In 2016, we started using laser microdissection. Here we collect the epithelium and also separate from connective tissue. In 2018, we investigated, we mapped primary tumor, focus on invasive tumor front and inner tumor. In 2022, we advanced to study the lymph node, the cells, non-malignant and malignant cells. I'd like to go deeper to explain a little bit what we evaluated in this study conducted by Carolina Carnielli, where we isolate malignant and non-malignant cells from the frontal of invasion and the inner tumor. Because we know that the inner tumor, there are processes associated with the tumor growth and maintenance. In the front of invasion, we can review some potential candidates associated with the tumor spread and metastatic capacity. And as I mentioned before, the metastasis, the lymph node of the patients are the main associated with the prognosis of the patients. At this time, we have already tried to evaluate a small number of cells. Of course, we have much less number of cells of proteins identified, but we evaluate small and large neoplastic islands. In 2022, Adriana, we evaluated several microenvironments from the patients, biofluids, but also we based our first experiments based on the spatial proteomics, where besides we evaluate the primary tumor, we advance evaluate the metastatic lesion, evaluating the proteome of non-malignant and malignant cells. And more recently, besides we dedicate our team to evaluate the progression of the head and neck cancer and oral cancer, we also decided to understand the initiation of the cancer based on spatial proteomics. So we evaluated both epithelial precursor lesions and normal oral mucosa to understand proteins that can be associated with the initiation. For that, we collect both the epithelial cells and also the cells from connective tissue in the oral normal mucosa, in premalignant lesion, and also in oral cancer, focus on the invasive front, both malignant area and non-malignant area. So over the years, the spatial proteomics have been helping us generating a lot of hypotheses, but also to move to hypothesis testing and to evaluate potential candidates and prioritize potential biomarkers and therapeutic targets through the validation and development of clinical assays, and also to explore potential therapeutic targets. To define and to prioritize and select the candidates that we choose from spatial proteomics to the next phase, we have been considering the clinical pathological association. This is one of the main features that we have considered to select the candidates, but also combined with machine learning classification to evaluate omics data from public databases, biological processes, the potential of the proteins to be druggable, and also the network, the relationship of the proteins within each other. And then we select potential candidates to be tested in the several experiments. And now I would like to show you how we can develop some initiatives for clinical translation from spatial proteomics. As I mentioned, we verify the candidates and then we plan to develop and validate and develop clinical assays. we have been testing at least two strategies. The first one, the major one, is to evaluate and verify and validate this candidate through target proteomics using both selective reaction monitoring and parallel reaction monitoring. But also thinking about how can we give access to the patients and to the professionals, we try to develop lower cost biosensor, fast and accessible biosensors. Of course, that we have been using the biosensors guided by the mass spectrum-based target proteomics. So I will show a little bit about this result we have conducting to use this potential phase to validate potential candidates. In this study conducted by Daniela Granata and Carolina Carnielli, in the first phase, we verified the potential candidates that were retrieved from the mapping of the primary tumor, selecting candidates associated with the prognosis, the worst prognosis of the patients. We select this candidate and we verify by select reaction monitoring using relative quantitation and machine learning classifiers. In that time, using 4 patients. And now we are trying to validate using absolute quantitation to estimate the candidates that classify the patients regarding the presence and the absence of metastasis. This is an ongoing project. So we have been using, as I mentioned, select reaction monitoring. We have adding synthetic peptides to estimate this concentration of this peptide of this peptide in saliva. This is the prognosis signature that was retrieved from spatial proteomics, and then classify the patients using machine learning methods. This is the partial result that we have been evaluating the curves of these different concentrations of this peptide. And this is more than 60 samples that we have already evaluated these peptides consider the 3 different peptides. And then we reached a partial number of samples, but we have already shown potential to discriminate the patient with and without metastasis, achieving the area under the curve 0.92. And now thinking about how can we give access to these results and also to this test to the patients, we have been proposing developing these biosensors based on the studies that we have conducting using mass spectrometry guided biosensors. In this case, we are testing immobilizing antibodies that recognize these proteins and testing saliva of patients without and with metastasis. For that, Luciana Trinalbano, she built this biosensor, and you have several specific features. And then we finish this part of the experiment also using machine learning classification. And by using 60 saliva samples, we have very good results regarding the lower limit of detection and quantitation. And after testing the 34 machine learning classifiers, we select this signature, and that could be discriminated patients with and without metastasis with the area under the curve of 0.75. 76. Besides these 2 strategies that were originated from that first experiment, first study that we mapped the proteins in the primary site of the tumor, we also evaluate the metastatic site, as I mentioned before, that Ariane Bussolopes conducted these studies. And we observe potential proteins that can be associated with the metastasis of the patients. And after the classification by machine learning, we evaluated these potential proteins associated with metastasis by using the flow cytometry. And these results indicate these 2 proteins that show a lower abundance in the samples and in the blood. In fact, mainly in neutrophils and lymphocytes, indicating that these proteins can be associated, the abundance of this protein can be associated with metastasis. Besides that, it opened another way to study the progression of the disease. As we observed in this study, that several of these proteins were associated with alternative splicing. And then we enter in another part of the study that it was the first time that we enter in this strategy to evaluate the potential modification of proteoforms by this. We don't know yet what's the mechanism. We are trying to develop some studies to understand what the mechanism or if the proteoforms can affect or are involved in the progression of the disease. In fact, in these studies, we tested it, and by using these spatial proteomics and also including, besides the cancer sites, we include the premalignant lesion and also the normal mucosa. We evaluate that we compare the proteins identified in the epithelial site and the connective sites, and we observe different abundance proteins compared to the normal oral mucosa in premalignant lesion. And when we evaluate the main process associated with the proteins, and for our surprise, we identify several proteins associated in the alternative splicing, in RNA splicing, mainly in the connective tissue. And because of that, we decide to advance to proteogenomics to understand whether the tissues, the progression of the disease were associated with the proteoforms. Then we perform the bulk RNA sequencing of normal oral mucosa premalignant lesion in oral cancer, and combine it with our RNA from a database with the UniProt database, including canonical and non-canonical sequencing. We evaluate our spatial proteomics data, try to retrieve splicing-derived proteoforms. And showing here, because of the time, on the results of connective tissue, we observed that 54% of these proteins are associated with cytoskeleton. We found 14 alternative splice-derived proteoforms when we combine both RNA, our database, or our databases built by our RNA-seq and UniProt. And interesting to note that several of these proteoforms are associated with cell immune populations, such as monocyte, T cell, dendritic cells, and B cells. Besides that, not only the connective tissue show these 14 proteoforms, but we can observe that 4 of these proteoforms are associated with the progression of the disease. So we could retrieve that these 4 proteoforms are different abundance when we compare normal oral mucosa, premalignant, and oral cancer. And this also, we evaluated their association with the pre-mRNA processing. So next, all these results, the splicing-derived proteoforms will be functionally characterized, and this promising result will support a deeper investigation of proteoforms, both using middle-down and top-down proteomics. And then now, as I mentioned before, the studying biofluids reveals several potential candidates as diagnostic and prognostic biomarkers. But we have been using bulk biofluids. And now we decide, not now, but 1 year ago, we decided to move to single cell proteomics, try to retrieve potential candidates as biomarkers, both diagnostic and prognostic biomarkers. So for this to enter in this area, Eloisa Monteiro and Jackson Miyamoto, they have been working a lot to establish this methodology in the lab. So first, we started to establish the benchmarks using the standard protein digestion and cell culture, both fresh and cryopreserved. So have to change and improve several controls that we had in the lab over the years. So establish specific instrument conditions, system suitability, sample quality controls, and daily routine of the projects. So now showing a little bit about our data, I will show you the standard HeLa protein digest and also the results we obtain using single cell HeLa benchmarks. So we, as to meet the international numbers, we follow the same approach they use, perform a curve of these peptides in the column, and then evaluated this data by meta evaluation, direct DIA with 3 replicates, and direct DIA with all the files, both at protein groups and peptides. And then we observe the coefficient of variation showing MS1 better than MS2 and covering 5 orders of magnitude. For the single cell benchmark, we improve a lot our contaminations, and we evaluate both fresh single cell and also cryopreserved because we want to move to clinical proteomics at the bedside. So we also observed a high numbers, and we have been meeting the international numbers both at protein groups and peptides. And we also have a very good coefficient of variation. And it's interesting here that we also achieved 6 orders of magnitude. Of course, in the beginning, I will not go through all these challenges, but it's important to mention here that we have many challenges. We can spend the whole day tell you about how many strategies we have to improve and to change to meet the international numbers. And I'd like to share with you, for example, here, when we have met the numbers, and then we have to change the column, we have to include a specific heater in the column, so it's not possible to have the heater in less than 2 or 3 months in Brazil. So Romania and Vanderlei, they solved that. They created an heating, and then we met the numbers. So it's important to mention here to enter in every project, but specifically for single cell proteomics, you have to have a lot of creativity and resilience to be able to run and have quality in the data. So then after meeting all the benchmarks numbers, we decide to move to application of this knowledge to biofood. So we decided to evaluate both saliva and tears from a pool of individuals. So we have for this analysis also, I didn't mention at the beginning, but we have the CELE-1 and also Astral combining with Vanquish New, and also have been performing the data analysis using both main spectronauts and develop some in-house strategies. So for this analysis, in the beginning, when you have a very we analyze saliva and tears, we have very difficult because it is an heterogeneous population. So as soon as we run all the raw files, we notice that when you have a large global difference, we increase a very low quality of peptides in the on-the-fly spectral library. So we decide to filter some of these bad raw files that we can see here. For example, it is a good data in this from saliva and from tears. So a data like that in this chromatogram, we need to filter out. And then we, after learning a lot how to process the data, we start with a lot of numbers, but then we have to exclude some raw data to improve in the end our number of protein groups and number of peptides. And then you can see here that you have both the cells from saliva and the cells from tears, that we can see specific clusters that are associated with specific biological processes, both in saliva and also tears of these 10 patients. And also it was interesting that you can also observe some specific clusters and some cluster markers that were retrieved and able to show this separation both in saliva and tears. But we need to perform the cell annotation. And to perform the cell annotation, we have also to have a very high creativity to try to process this data and to perform the cell annotation using proteomics and not transcriptomics. And for that, we use our spatial proteomics to select the epithelial markers and to exclude, based on the public database of Hickman 2017, to annotate the immune cells. And then after that, we could annotate the cells based the cells based on the clusters, indicating that we have both epithelial cells in saliva and mainly granulocytes, monocytes, and NK cells. In tears, we have mainly granulocytes. But then after learning the benchmarks, learning using the samples, using pool of samples, then another challenge would be to collect the samples of the patients in the hospitals. So we have to also repeat the experiment several times to be able to select the best cells or that the cells to have high integrity to be able to be isolated by the cell one. So after several experiments, we decide to sort the cells before cell one and select several strategies to select the best cells, or I can say not the best cells, the cells with high quality to be able to be isolated and then follow the workflow. So we also established some quality controls to improve the quality of the analysis and give us the confidence that the analysis work correct and we are working in the maximum of our capacity. So we are adding single HeLa cells in the patients of the well plate of each patient. Important to mention that is one patient per condition. And here, as you can see here, we have 5 single HeLa cells per plate, and we can see that some cells that are grouped here, but there are some cells that are not. And here I can show you that we collect more or less 135 cells for each patient. And then regarding the control patient, we can see that there is a correlation of the number of protein groups regarding the diameter of the cells. And it also occurs in the patients without metastasis here. But the patient with metastasis, we cannot see this correlation. But interesting to note that also we have to exclude more cells. And here I'm showing the results of the protein groups of these cells collecting from one patient in each graphic. And here I can see that in this case, we use this library search that we have 10 cells in each plate of the patient. And we excluded the CREP entries, and also for this graph, we exclude cells that have less than 100 protein groups. And to annotate the population, and also to evaluate whether what proteins are in these different patients and what proteins can be potential markers. Although we have one patient per condition, patient control, patient without metastasis and patient with metastasis, we can see that we have more or less separation of this patient, showing more control cells here, patients without metastasis, patients with metastasis. And we can see here more specifically these clusters based on Leiden method, that we can retrieve from these cluster proteins that could be able to select or cluster markers that could be highlighted by the proteins identified in patients with oral cancer without metastasis and patients with oral cancer with metastasis. Finally, I'd like to mention that we were still working to improve single-cell proteomics workflow for analyze the biofluid-derived clinical samples. And our next challenge, continue several challenges, and implement untargeted spatial single-cell proteomics to investigate cell-to-cell communication at the invasive front that can understand, can give you some insights about the cancer initiation and the progression, and retrieve more potential candidates as diagnostic, prognosis, and therapeutic targets. With that, I'd like to thank you very much all the patients that provide their samples to our studies. I'd like to thank all the funding agents, and also enjoy this opportunity to invite you to join the proteomics workshop. We'll have the hands-on this year. Thank you very much for your attention.
Thanks a lot. Thanks a lot, Adriana. Wonderful, wonderful presentation. I've seen there are also a lot of participants that followed your presentation. Can you hear me well?
Yes, yes.
Can you hear
Thank
me,
you very
Adriana?
much, Joseph.
All right.
Yes.
So I have seen that there is.
I should stop.
Yes.
Can I stop
please.
my?
Yes. Okay. Yes. So I've seen already that there is a question in the chat. The question, of course, starts with wonderful work, Adriana. How many cells did you use per condition in the single cell experiment? So as referring at the last topic you present.
I'm checking here. It's in fact depend on the experiment. When we did the pool of the cells from 10 individuals, we use for example, for saliva, single cell from the healthy volunteers, we use more than 100, and for single cell, we use 150. But for the patients, although we use, in fact, we sort a lot of cells for when we use the sorting to select the cells from the patients. But then we isolate the cell 189. But then after running in the samples in Astral, we will have to exclude some cells that has less than 100 protein groups. So in the end, we consider, for example, in this last study, for example, for control patients, we use 123 cells. For patients without metastasis, 100 cells, and patients with metastasis, 84 cells. But in the beginning, we analyzed 189, and then filtering in cell 135. I don't know if it's clear, Fernanda. But yes, it was not a fixed number. And it's one question that we always asking us, what's the best number of the cells? It's not easy to know, because depend on what you are comparing. So it's not I think we need to work more, but at least using this number of cells, that's more than 50, that's at least a minimum 50 that we have been heard, but we have been hearing. I think with this number of cells, we could discriminate patients, control patient with and without metastasis.
That's amazing sensitivity. Just that 85 and 100 cells. So how could other people reproduce your data? So do they need the exact instrument you are using, or is that the technique you are using?
Yes, and another thing about patients, because we have a lot of challenging to define the best condition to collect the cells. It's something that we have to train the dentists that are collecting the cells in the hospital, because we decide that we change a lot of the protocol. We cryopreserve the cells in the hospital, and then we transport to Campinas, and we store the cells in -8. And then in the other day or 2 days later, we start with the protocol of sorting the cells. So I think our plan is to describe it in the very details our protocol, try to help the users or other people to use the similar protocols. We test a lot of protocols here, and then I think now we could reproduce along the patient's collection.
That's fantastic. Thank you. That's another question from the audience. Says, amazing work, Adriana. How did you differentiate between metastatic and non-metastatic cells?
Okay. In this case, regarding the spatial proteomics, this is in the spatial proteomics. Yes. In this case, we name as metastatic and non-metastatic because we collect the metastatic cells in the lymph node. So the cells are in the lymph node. In non-metastatic cells are the cells that are not, most of them are non-immune cells, but also fibroblasts, and all the laser microdissection were performed by pathologists. So it's something, but then of course you have some contamination, and then in the annotation of the regions, for example, in the last case that we need to annotate the biofluid cell population, we combine the data where we combined what Hickman et al. Found, consider this, they sort the immune cells, and we exclude all the potential proteins that could be clustered as immune cells, and we separate as epithelial cells. So we really need to compare this public data and our data to select the non-malignant and malignant cells.
Thank you. This tremendous amount of work.
Yes. So, Adriana, there is another question from the chat is regarding machine learning. So, which machine learning model did you use to train the data for the spatial proteomics data?
I don't know if the postdocs are here. I tell them that they could answer the questions. So the people bioinformatician people from my team will answer your question there.
Great. So then I have a question regarding the first part of your presentation, when you show the validation of some markers, and I could see that one of the peptides that you were using for validation was related to collagen 6, right?
Mm-hmm.
And I was thinking in your study, when you analyzed the invasion front and the primary site where the tumor is, if you have detected some changes in the extracellular matrix organization, that because I mean, and if you have thought about using, I mean, at least some novel therapeutic strategy to see if they work to reduce this metastatic potential, working on modulating the matrix, the extracellular matrix.
In the study of Ariane, we evaluated, we compare, Joseph, the cells, the malignant cells, the primary tumor, and the malignant cells in metastatic site. we have evaluated the ability of the matrix. In fact, we quantify some proteins in the matrix, and we noticed that the proteins associated with epithelial mesenchymal transition. So we have this, and we did notice that there is a modification in the extracellular matrix, and also within the cells that change the matrix. And another thing that's important to say that we decide, I did not show you here, but for this study that we analyze the normal oral mucosa and pre-malignant lesion and the progression of the disease in the front of invasion, we also identify fibroblasts that has been associated with the progression of the disease. And fibroblast is really involved in the modulation of extracellular matrix. So for this, I did not show here, but we have another study that we isolate the fibroblasts from the tissue, and we also evaluate the extracellular vesicles of fibroblasts that was modulated by the tumor microenvironment. So I think there is some specific strategies that we can be used here to modulate the extracellular matrix. And we really want to improve and try something that can, for example, we have another proteins that we study in extracellular matrix, which is a proteoglycan. And this proteoglycan is associated with the progression of the disease and show high abundance in the oral cancer. So I think there is a lot of ways that we can modulate and try to modulate the progression with the inhibiting some processes. Even in fact, in this case, that the proteoglycan, we need to inhibit the interaction of this protein with the other two proteins associated with the progression. This is one of another study that we are conducting regarding the extracellular matrix.
Yeah, that's great. Thinking also about again this mix, did you see some changes in MMPs as well? Because normally they are very well studied in cancer. So I want to ask you if also in your data you see some changes in MMPs that could be also related to remodeling of the extracellular matrix.
Yes, I do not remember a specific metalloproteinases. We focus a lot in the immune cell population because of this opportunity to explore the alternative splicing. But yes, I can check and we can discuss later about the specific for the metalloproteinases.
In the Q&A, that's it. Okay, in the Q&A, that's a comment by Fernanda Salvato. I love that Adriana walked us through the process of how much optimization is necessary in collecting viable cells to have reliable results in single cell experiments. Thank you. And there's a
Thank
question.
you, Fernanda. Thank you, Fernanda. You know that Fernanda helped us a lot with all the process. So thank you very much, Fernanda. Your name is there in the acknowledgement. So thank you, Fernanda, for your help.
There's a question from Gilberta Domento. How many protein groups do you identify in a HeLa single cell experiment?
So in the HeLa, my dear Gilberto, let me check here. So we tested two protocols using the single cell HeLa. One is fresh, it would be the gold standard, but also we test the cryopreserved because of the collection of the cells in the hospital. So we identified, for example, we have the blank, you have method evaluation, and consider that the search using library search with 10 cells, we identified 4,921. The cryopreserved cells, and using match enhancer, we identify the 4,901. So it's a great number. At least we are trying to to meet the international numbers, and it's more or less the same. But then it's important to mention that we really want to use the cryopreserved data, and it is a little bit better, in fact, than the fresh, because the cells are immediately stored in the FBS. There is one more question here.
So one more question, yes, is regarding single cell proteomics, not in Astral, but in other instrument, also from the orbiter. Do you think it's possible?
Yes, definitely. I think the main challenge is to have a very quality cell preparation. So I think something that we really need to improve, to have a very quality preparation, isolation of the cells, and preparation without transferring a lot the digested peptides. So something that it's really possible to run in other instruments.
Great.
Fernanda is yes, Fernanda, we are, as you know, working in the monopeptides. Yes, we are learning. We have only one Astral. If you have two Astral, probably we will run faster.
That would be great. All right.
Yeah.
So I think there are.
I have
Oh,
one more
yes,
question.
shoot.
So you have a big operation. You also, in the very beginning, you indicated that it's open to external investigators as well. So how do you charge? How do you bill? How does it work?
No, here the users are not charging. They can apply their project, and the external committee recommend their projects, and then they come here and do the whole processes with the specialists and the team that we have. It's how the national laboratory works here in Brazil.
Oh, okay. So the reagents are also paid by you?
Yes.
Everything, the instrument time. And you
Yes.
also indicated that people will come, the students will come to prepare samples and also learn about the instrumentation, everything for your teaching project.
Yes, but they do not operate direct. They have the specialists that help them, but they come to learn how to prepare the samples. They can participate in the injection of the samples, and then they learn how to process their data.
Wow. So how many people can you accommodate? Like if you have 10 people all want to come this week, you be able to?
Yes, we have users every day here, but in each year we accept about 100 proposals. We have yes, it's
So
100
that's
proposals.
within Brazil, right? You don't accept proposals from the US?
Yes, we
You
accept.
do.
Yes.
Okay, thanks.
All right. So I think we can assess. There is one last question before closing this session. So Adriana, is regarding if you found, is from Michelle, if you found some overlap between biomarkers in the bulk analysis and single cell.
We still have this preliminary data. We are continuing this study regarding this alternative splice, and that we are trying to improve. We did not check yet because most of these proteins that we select as potential marks are extracellular proteins, but we are on the way. We are still learning about the results, and I don't know if you will find that. In fact, we have these clusters that were selected based on the lighting method. And then probably we need to still work and learn more. But until now, for these proteins that we evaluated, it is not the same as that we have been identifying in the biofluids, at least the ones that we are in more advanced phase of validation. So they do not match yet.
All right. I can see there are still questions coming in. Let's get this last one. We are still a few minutes left. So is from Professor Gilberto. Adriana, he is asking to confirm if you use the combination of SELEN1, EvoSep, and Astral for your single-cell proteomic experiment.
Gilberto, for this the EvoSep one is coupled with Exploris 240. For the Astral, we have the Vanquish New, and then we have this workflow that now for the patient samples we have the sorting in S8 flow cytometry, and then we go to the SELEN1, then to Vanquish New and Astral. In that time, we decide to not, because we need to increase the throughput in the Exploris 240, and then we decide to install EvoSep LC in Exploris instead of Astral.
All right. I think we can close this webinar. It was great, Adriana, having you here. I thank Shisha for co-chairing with me this session. HUPO office, Emma is behind, helping a lot with the infrastructure. Thank you, Emma. And also, of course, HUPO and the ETC committee for giving us this opportunity today. Thanks a lot, and we can close the session. Thanks, Adriana.
Thank you,
very much.
Adriana. Thank you, everyone.
Thank you. Nice to meet you. Thank you. Bye-bye.
Bye.